Hello, and welcome back. So this is our video lesson on 2.4, transport across the membrane. If you are ready, let us jump straight into the lesson. So this is still part of chapter 2, cell structures and functions. A quick overview of what we have in this subtopic is we will overview the various types of transport mechanism across the membrane, including passive transport and active transport. We will also cover the concept of water potential and calculation of water potential, and this will be included in osmosis. Okay, so when we talk about transport mechanisms across the membrane, how we bring things in or out of the cell, it can fall into one of two categories, which is passive transport or active transport. Now, I'm sure as we are learning each types of these transport, you will start to compare them as we go along. So, apa-apa perbandingan yang kamu dapat fikirkan, okay? Coba ingat dan nanti coba untuk banding bezakan sambil kamu tulis. Okay, when we learn about passive transport, we will talk about simple diffusion, facilitated diffusion, and also osmosis. When we talk about active transport, we will talk about the sodium-potassium pump and also bulk transport. In bulk transport, we will look at endocytosis and exocytosis. Okay, let us begin with passive transport. What's passive transport? Passive transport refers to the movement of molecules across semi-permeable membrane from a region of high concentration to a region of low concentration. These molecules will move down the concentration gradient, or we can also say they follow the concentration gradient, or the molecules move along the concentration gradient. And they do all these without any energy requirement, or ATP. Dalam arti kata lain, bahan bergerak dari kawasan yang banyak bahan tersebut ke kawasan yang sedikit bahan tersebut. Tapi bila kamu perlu bagi definisi, sila gunakan key terms higher concentration, lower concentration, semi-permeable membrane. Kalau paling bagus, sila gunakan term concentration gradient and without energy requirement. So there are three types of passive transport, which we will look at one by one. The first type of passive transport is simple diffusion. Simple diffusion is the movement of molecules or substances across a semi-permeable membrane from a region of high concentration to a region of low concentration without energy requirement. Kalau kamu mau bagi definisi, sila bagi definisi mengikut ayat ini. In simple diffusion, we do not require transport protein. So, dia akan terus limpas phospholipid bilayer. Tiada siapa-siapa tolong dia. Molekul-molekul yang boleh melalui simple diffusion adalah molecules that can undergo simple diffusion are lipid-soluble molecules or non-polar molecules or hydrophobic molecules, such as estrogen, progesterone, testosterone, vitamin A, vitamin D, vitamin E, vitamin K. Okay? Dia akan terus limpas phospholipid bilayer. This is possible because the area in the middle of the phospholipid bilayer over here is hydrophobic. So, benda yang hydrophobic senang sahaja terus lalu. Benda yang hydrophilic tidak boleh lalu. Besides that, another type of molecule can also pass through, which are small, non-polar, and uncharged molecules. So things like oxygen and carbon dioxide can diffuse in and out of the plasma membrane freely. Okay. The next type of passive transport is facilitated diffusion. Facilitated diffusion is the movement of molecules across a semi-permeable membrane from a region of high concentration to a region of low concentration through channel and carrier protein without energy requirement. So, tadi dia terus melalui phospholipid bilayer. Ini dia mesti melalui protein. Protein itu adalah sama ada channel protein ataupun carrier protein. Let's use the example of water. Water can come in and out of the cell through facilitated diffusion, but they go through this protein called aquaporins to facilitate rapid movement of water molecules across the membrane. Other molecules that enter or exit the cell via facilitated diffusion are polar or hydrophilic molecules, and these are usually larger molecules such as glucose and amino acid. Other types of molecules that need to undergo facilitated diffusion are charged ions, such as sodium, calcium, chloride, this thing, and also potassium. Okay, so as we can see from this diagram here, if you have a channel protein, these usually allow charged substances such as ions. Okay, macam begini, dia boleh terus lalu. Carrier proteins also don't need energy, but they have a specific binding site. The substances will bind on the site with higher concentration, the protein will change shape, and it will allow the molecules to come out of the binding site on the other side of the membrane. So, sebelum itu molecule masuk, dia punya bentuk lain. Bila itu molecule sudah melekat, dia punya bentuk berubah. Bila bentuk dia berubah, molekul tersebut boleh melalui dan masuk ke dalam cytoplasm. Okay, and we are moving on to the third type of passive transport, which is osmosis. Osmosis has a slightly different definition. Kalau kamu kena minta untuk bagi definisi untuk osmosis, sila gunakan definisi ini. Osmosis is defined as the net movement of water molecules across semi-permeable membrane from a region of high water potential to a region with low water potential without energy requirement. Kalau kamu perlu bagi definisi osmosis, sila ingat keyword ini. When you give the definition, you must have these keywords, which are net movement of water, semi-permeable membrane, high water potential to region with low water potential without energy requirement. Perlu ada keempat-empat keyword untuk mendapat markah definisi. Okay, so let us talk about the concept of water potential. Apa bendakah water potential? The water potential of a solution is the tendency for water molecules to leave the solution by osmosis. The more concentrated a solution, the lower is its water potential and the smaller for tendency of water to leave the solution. Okay, water potential ni kita boleh gunakan simbol psi, which is this thing that looks like garpu. Benda yang nampak macam garpu ini adalah simbol untuk water potential. Kalau air itu adalah air suling ataupun pure water, distilled water, no solute whatever, then it will have a water potential of 0 kilopascal. Okay, 0 kPa. This is the maximum water potential. Tiada water potential yang lebih tinggi daripada kosong. Memang paling tinggi adalah kosong. Kalau dalam air suling itu, saya letakkan sedikit garam. If I put a little bit of salt in my distilled water, it will become a dilute solution. It will have solutes. Remember, solute is the garam, solvent is the water. So in this one, if you have even a little bit of solute, your water potential becomes negative. This example, we have a dilute solution with negative 200 kPa. If you put even more salt in the solution, kamu tambah lagi garam dalam solution itu, kamu akan menurunkan lagi water potential solution tersebut. You are going to further lower the water potential. So if I add more salt, my concentration will become negative 500 kPa. Remember, water molecules move from a region of high water potential to a region of low water potential. So bila kita letakkan dia begini, air akan bergerak daripada kosong ke 200, negatif 200 ke negatif 500. Water potential or site with more negative value has more solute and thus lower water potential. Bila solution itu ada banyak garam, banyak solute, air akan pergi ke tempat dia. Air tidak akan tinggalkan dia. So that is what is meant. Water has less tendency to leave the solution. Okay. As you notice, water potential of a cell is a negative value because the cell will always contain solute. Dalam sel kita sentiasa ada benda yang terlarut. These solutes make water potential become negative value. If you are looking at an animal cell, the solute potential that we give is the water potential of the cell. If we look at plant cell, you need to use this formula. Okay, formula yang kita ada di sini for calculation of water potential is only for plant cell. The formula is water potential of the plant cell is equal to solute potential plus pressure potential ataupun psi equals to psi S plus psi P. Okay. Solute potential is the potential of a solution to gain water. Value of solute potential is always negative. If solute concentration increase, solute potentials become more negative. Ini lebih kurang apa yang saya cakap tadi. Bila kita tambah garam atau kita tambah solute, dia semakin negatif. Okay, solute decreases water potential. Solute lower the water potential by diluting the water molecules. Next is pressure potential or psi P. Pressure potential are hydrostatic pressure that is exerted on a water in a cell. It can be positive or negative relative to atmospheric pressure. Macam mana kamu mau tahu positif atau negatif? Kamu baca sahaja soalan dengan bagus. Just read the question carefully. You will know if it is positive or negative. Next, we are looking at the effects of solutions of different tonicity on cells. Dalam arti kata lain, kalau kita ambil plant cell dengan animal cell, kita masukkan dia dalam larutan garam yang berbeza kandungan garam dia. Apa terjadi kepada sel-sel tersebut? What happens to each type of cell when we put it in a hypotonic, isotonic, or hypertonic solution? I'm sure you've seen this diagram before, but I'll just go through it again. Okay. Hypotonic solution means less. A hypotonic solution has a lower concentration of solutes than the cell cytoplasm. So that means kamu punya larutan garam itu kurang garam lah. Okay. So water molecule will diffuse or move into the cell by osmosis because the cell will have lower water potential. Okay, when this happens, in an animal cell, the animal cell will become lysed ataupun burst. Dia meletup sebab lampau banyak molekul air masuk ke dalam dia. Dalam plant cell, in a plant cell, the plant cell will not burst, but it will become turgid. Dia macam mengeras, bukan mengeras lah, like tegang. Okay, permukaan sel itu tegang, turgid. If you put it in an isotonic solution, iso means same or equal. An isotonic solution has the same concentration of solutes to the cell cytoplasm. So, concentration di luar sel dan concentration dalam sel adalah sama. So, the water molecule will diffuse in and diffuse out of the cell at the same or equal rate. Bilangan water molecule yang masuk dan bilangan water molecule yang keluar adalah sama. Jadi, kamu punya animal cell tiada perubahan dari segi bentuk dia. Your animal cell will be normal shape. Your plant cell will also be normal shape, but it will be a little bit flaccid. Flaccid dia macam lenyek, lembut sikit. Okay. And if you put your cells in a hypertonic solution, hyper means more. A hypertonic solution has higher concentration of solutes than the cell cytoplasm. That means the water molecule will diffuse or move out of the cell by osmosis. When water molecules are moving out of your animal cells become shriveled or crenated. If this happens to your plant cell becomes plasmolysed. Plasmolysed means dia punya cytoplasm mengecut, tapi dia tidak terus mati lah. Cuma dia punya plasma membrane akan tertarik sedikit daripada cell wall. So if you have to draw a diagram of a plant cell that is plasmolysed, show a bit of space to illustrate that the plasma membrane pulled away from the plant cell wall. Okay, shouldn't have too much problems there. And so far we have gone through simple diffusion, facilitated diffusion, and osmosis. Let's move on to the next segment, which is active transport. So just now we were talking from high to low. Now it is the opposite. In active transport, molecules move from a region of lower concentration to a region of higher concentration. The molecules will move against the concentration gradient. So, molecule bergerak menentang concentration gradient. This process requires energy in the form of ATP, and it will involve carrier proteins. One common example of active transport is the sodium-potassium pump. This carrier protein is used to maintain the concentration of sodium and potassium ion across the membrane. Normally, when we compare the inside of your cell, the cytoplasm, to the outside, the extracellular fluid, bila kita bandingkan, kamu boleh nampak there will be low concentration of potassium and high concentration of sodium outside of the cell. Inside the cell, there is low concentration of sodium, high concentration of potassium. What does sodium-potassium pump do? Sodium-potassium pump ensures that this concentration gradient is maintained by pumping sodium ions out and pumping potassium ions in. Sodium-potassium pump, fungsi dia adalah keluarkan sodium, masukkan potassium. Mari kita tengok macam mana dia melakukan benda tu. Okay, so how does sodium-potassium pump work is we start with the plasma membrane, of course. At the plasma membrane, we look at the pump. You will have 3 sodium ions in intracellular cytoplasm bind to the sodium-potassium pump. So 3 sodium ions will go and stick here. And then you will use the ATP. To use energy from ATP, we need to break the ATP. Macam kamu mau kasih patah KitKat, kita kasih dia. Okay, so you take the ATP, you remove one phosphate group. One phosphate group will be transferred from ATP to the sodium-potassium pump. Kita kasih melekat dia di sini. Itu ADP, kita buang. This causes the sodium-potassium pump to change its shape or conformation. Remember, the word conformation is just fancy science word that means shape. So this causes the sodium-potassium pump to change its conformation. The 3 sodium ions are released to the extracellular fluid outside the cell. So 3 sodium ion ito dibuang keluar. Next, 2 extracellular potassium ions bind to the sodium-potassium pump. So 2 K plus ion dari luar akan melekat di bahagian ini. This causes the phosphate group to detach from the pump. Tengok, bila dia sudah itu potassium ion melekat di sini, fosfat akan tercabut. Bila fosfat tercabut, sodium-potassium pump will return to its original shape or original conformation. Dia akan balik kepada bentuk asal. When it returns to its original conformation, the two potassium ions are released into the cytoplasm and the pump is ready to bind 3 sodium ions again. Dia akan bebaskan dan dia sedia untuk repeat sekali lagi. So the cycle will repeat again. If you're unclear with any of the instructions in the sodium-potassium pump, please feel free to ask me or a classmate. Okay, so that is it for the sodium-potassium pump. Next, we shall talk about bulk transport. Let's talk about bulk transport. Bulk means secara besar-besaran ataupun secara pukal. When we talk about bulk transport, we are talking about the movement of large molecules such as proteins and polysaccharides, large particles, large amount of molecules across the membrane, sometimes even an entire bacteria. Bulk transport is involved in the formation of vesicles or vacuoles. Okay, dia akan melibatkan pembentukan vesicle ataupun vacuole. And there are 2 types of bulk transport. Endocytosis, which is bringing things into the cell, and exocytosis, which is expelling things out of the cell. Okay, first type, endocytosis, bringing things into the cell. Endocytosis is a process where a cell transports macromolecules, materials, or substances in bulk by forming vesicle or vacuoles from the plasma membrane. It involves the invagination of the plasma membrane or the membrane fold around the material outside of the cell that pinches off to form vesicles or vacuoles. Dalam arti kata lain, plasma membrane akan buat begini. Okay, bila itu bahan yang kita mau kasih masuk dalam sel, masuk dalam ruangan yang plasma membrane buat itu, dia akan tutup bahagian atas, pinch off, dia macam tarik, ya picit, begitu. Sampailah dia membentuk satu vesicle ataupun vacuole. This entire process will of course involve ATP and energy. And there are 2 types of endocytosis. 2 cara kita punya sel membawa bahan masuk ke dalam cytoplasm, which is phagocytosis and pinocytosis. Phagocytosis is also called cell eating, ibarat sel kita sedang makan, and cell drinking. Pinocytosis is cell drinking, ibarat sel kita sedang minum. Okay, so let's talk about phagocytosis or cell eating. It's a process where living cell is taken up or engulfed. Sometimes it is living thing, sometimes it is just a normal solid particle, sometimes it is a bacteria. So this involves formation of vacuoles such as food vacuole. So plasma membrane akan memanjang begini, dia akan tangkap benda yang kita mau makan itu sampailah dia membentuk food vacuole, ataupun nama canggih untuk food vacuole adalah phagosome. Selepas ini, phagosome ataupun food vacuole itu akan digabungkan dengan lysosome. This food vacuole will be fused with the lysosome, and the hydrolytic enzyme from the lysosome will digest the material. Enzim dalam lysosome tu akan hadam atau mencernakkan phagosome, makanan yang kita dapat tu. Then the digested material will be absorbed into the cytoplasm. Apa yang sudah dicerna akan diserap oleh sel masuk ke dalam cytoplasm. Okay, so this is the process. from when you start eating to the vacuole to the kasih gabung and then dia sudah dicerna dan apa-apa bahan buangan akan dikeluarkan melalui exocytosis. Let's talk a bit more about phagocytosis. Examples of phagocytosis is when you have Kupffer cells in your liver, engulf or break down worn out erythrocytes and bacteria. So di hati kita akan ada sel khas yang mencerna darah merah, sel darah merah yang lama dan bakteria yang ada dalam darah. You also have phagocytosis by white blood cells. So your white blood cells like neutrophils and macrophages will engulf and digest cellular debris or foreign substances. Other things that are not in humans, for example, amoeba. Amoeba will capture its prey by extending the pseudopodia around the prey and then digest the prey. Okay, so these are all examples of phagocytosis, which is cellular eating. Okay, so we talk about eating, let's talk about drinking. So pinocytosis is a process where a living cell will engulf or take up material in liquid form, or it can also be a liquid that happens to have dissolved substances or solutes. Ini lebih kurang macam tadi, dia akan hasilkan vesicle juga. The vesicle will contain the dissolved solutes, and then the materials are absorbed directly into the cytoplasm. So, bahan-bahan dari vesicle ini terus diserap ke dalam cytoplasm. Dia tidak perlu menggunakan lysosom. Okay. If there is something our cell doesn't need or something our cell wants to export, then we use the second type of bulk transport, which is exocytosis. Exocytosis is a process of transporting materials or substances out of the cell in bulk by the fusion of vesicles or vacuoles with the plasma membrane. The contents in the vesicles or vacuoles are released to the outside of the cell. Dalam erti kata lain, ini dia adalah apa yang kita tunjukkan dalam gambar raja ini. The vesicle goes close to the plasma membrane. When it reaches the plasma membrane, the vesicle starts to fuse with the plasma membrane, forces everything out and out and out and out. What kind of material is transported via exocytosis? Examples of materials that are transported via exocytosis are enzymes, hormones, waste products, and mucus. Enzyme, hormon itu mungkin adalah enzim dan hormon yang dihasilkan oleh sel kita yang perlu dihantar kepada sel ataupun organ lain. Waste product, sometimes your cells produce waste product, bahan buangan, dia akan buang melalui exocytosis. We have specialized cells that produce mucus, and those mucus will be transported out of the cell via exocytosis. All of these will require energy in the form of ATP. Okay, so examples are secretion of enzymes from pancreatic cells in your pancreas, secretion of hormones from endocrine glands, and also removal of waste products from the cell. Of course, you can definitely compare between phagocytosis and pinocytosis. Kamu boleh banding-bezakan antara cell eating and cell drinking. So some of the similarities is both are types of endocytosis, both involve invagination of plasma membrane to form a vesicle or vacuole. But you can also find differences. So examples of, excuse me, where's my head? Okay, so examples of differences are whether they are solid or liquid, involve lysosome, don't involve lysosome, involve formation of vacuole or vesicle, how are they digested and absorbed? Does it involve pseudopodium or not? Okay. What should you do now? You should look at the past year papers Okay, pergi website KML belajar, cari di mana. Cari soalan pasir, chapter 2, question number 10, and then you do all the questions. Okay, please do question number 10, A and B. Sebab ini cover both the active transport. Oh, no, this covers the passive transport only. Ya, nanti active transport kita boleh buat latihan lain. Okay, so that is it for this subtopic. I will see you again soon, probably on Thursday, and also with the 2.3 videos. Thank you very much for your time and attention, and I will see you again soon. Bye!